Hi, everybody. Welcome to this session. My name's Alyssa Hartman. I'm with the Artisan Grain Collaborative, and I'm excited to introduce Dr. Darcy Telenko, who is going to be talking with us today about disease management in organic systems. She will tell this to you as well, but please feel free to interrupt with questions. Raise your hand. We've got a mic for you that we can run around so that you can find out what you need to know on this important topic. I'm going to hand it over to Darcy.
Well, good morning. Again, as we move along, I'm presenting what I think of some of the work we've done, but if you have questions or other issues you want to discuss, feel free to discuss. We're open to different diseases if there's anything of concern. I do think if you have questions, just come to the mic, but if you want to raise your hand, I'll try to repeat them if you want to interrupt me that way. That way they can record it and have it for the future. As I get started, I just want to acknowledge the work that I'm presenting here is a collaboration between myself and Damon Smith and Aaron here at Wisconsin, and a lot of the work was supported by a North Central SARE project. Let's see if I can get this to go. As a little side note, if you're interested, some of the resources that we have here for Indiana, I have Indiana Field Crop Pathology website. It has some of these maps that I may share, and that information on Twitter. I only talk about diseases, so if you're interested in some of the things we see in Indiana, those are the things that we report. And then also, we work with the Crop Protection Network, which is a wide resource of various diseases. It may not all be organic-based, but just information on the various diseases, looking at disease cycles when we think about how do we best manage each disease and each crop. And so I thought I'd just go over some of the real standard principles that we think about when we're thinking about disease management, whether it's organic or conventional production. And some of those things, they remain the same until we get to the tools that we utilize, right? That's what changes and what we can use in trying to manage them. So the first thing always is understanding what is the target? What is our disease problem that's causing an issue? Maybe you haven't seen diseases and you're not worried about anything, but what is that disease? Can we detect it early? Can we make a confirmation it's happening? And then can we make an informed decision to do something about it to minimize the impact in that season? Many of our diseases, by the time we really see those disease symptoms, so if I think about tar spot, by the time I'm getting calls about that disease in Indiana, probably it's too late. And I can discuss more about that. So it's important to be out and scouting and monitoring for those diseases, keeping a field history of what's going on in those fields to understand this disease is one concern this year, maybe down the road it still may come back again. Understanding what diseases is where, so if we have sclerotinia in our field and one field that's really bad, we want to make sure we're not moving that disease to a new field and introducing it where it wasn't a problem previously. If we think about our control strategies, we can classify them in 4 areas. So exclusion, the idea is we're going to keep the disease off the farm. Some of our diseases, we may be able to do that. A lot of them are soil-borne pathogens. Maybe if they're not on your farm, if we don't bring in them, they may not become a problem. Some of the diseases we can't do anything about. They're moving in the air. We have spores moving around the United States. We can't really do anything to keep them out. We can also think about how do we reduce the inoculum. And that's kind of where rotation and tillage may come into play, is how do we minimize the amount of inoculum that's surviving year to year that may be available for when that crop comes back into the system. And then we can look at chemical and biologicals as well, as how do we incorporate them if the disease is of risk. So I look at an integrated disease management program. So we have these control procedures. The idea is to try to utilize the earlier stuff to then minimize our pesticide use, right? And so it's important to understand the pest's life cycle in order to target different control measures. So again, scouting is important to understand what disease we're targeting, and then understanding how that pest is moving in the system, where we can target different things. And I'm going to use 3 different systems. We'll talk about their disease cycle and where we might target management tools, both organically or conventionally, but we'll focus more on the organic today. So I look at it encompassing a system. So first, can we start with some host resistance? Can we identify the cultivars, hybrids, varieties we want to use that have higher resistance to that disease that is of concern? Can we also look at cultural controls, rotation and tillage? In a corn and soybean system, we really have a short rotation, where if you add additional rotational crops within that system, we lengthen that period from when that original crop, so let's just say corn is our target, we want to minimize, just to say tar spot. If we go at a longer rotation, we might reduce that initial inoculum that could start the disease. We also want to think about scouting and pest forecasting, so we have some tools for some of these diseases we're going to use that can help figure out if we need to maybe target a pesticide application in season if it's necessary, if there's a product available. We can also talk about biologicals, and I'm gonna get a little bit on some of the biologicals. That's where this project looked at, is some of these biologicals that have OMRI labeling. Will they be effective more in our row crops? And then, of course, chemical control if that's available. So again, exclusion, we're keeping that pathogen from an area. So that's why we have the USDA and APHIS and PPQ that are protecting our borders. If we don't have the disease in the United States, we don't want that new disease coming in. So that's some of the things we try to exclude. We may want to not move around nematodes that may be a problem. So there's things like that that we have in place to try to quarantine when we have new pests move in. We can also look at if we're not generally row crops, but disease-free plants, disease-free seeds. Are we starting with a clean base before not introducing a new disease? Again, you could look at your seed catalogs, identify those that are certified disease-free. If we have bulbs, again, making sure we start clean, not introducing that pest into the system. If we think about inoculum reduction, the idea is we want to reduce that initial pathogen propagule from that field. Whether it's removal or reduction of those plant pathogens from the area, maybe we can remove diseased plants. So for example, if I'm thinking outside, if we're talking about late blight, maybe we can pull on those plants and the tomatoes and remove them from the field. Maybe we can remove diseased tissue. A lot of these fall into other crops other than corn and soybeans, but again, some things that we can think of how we can reduce that initial inoculum. Again, rotation's probably our best way in corn and soybean systems. And then looking at chemical controls, so we have different fungicides that can be used. We have fungicides, bactericides, nematicides, very few nematicides. We have seed treatments for some of those. We have no chemicals to kill virus diseases. If we think of our fungicides, even if they're OMRI labeled, the protectants need to be there before the pathogen infects. Before that spore lands on that leaf surface, we have to have the protectant there. Our systemics will move into the plant, so even if a spore is landed ahead of the protection, they may be able to slow down the infection and reduce that disease that develops in the canopy. And then biopesticides. So this is a large group of products that are now coming available. They're naturally occurring substances. They may be biochemical things, microorganisms, substances produced by plants, and many of those are what we see that are labeled that have that OMRI listing. And so we recently put out a new publication looking at biopesticides. If we look at all the biological products that are available, I'm talking about disease management on this side. So I'm looking at the ones there, the biopesticides, microorganisms, biochemicals, anything that may influence the pathogen side of things. There's a whole list of other ones that may be used for nutrition and other things. That's not what I'm targeting with our work. We're looking at, can we introduce a microorganism with biocontrolling the pathogen, eliminate it from the system. And so if we look at the biochemicals, those are the natural occurring substances. Maybe they're plant extracts, neem oil, seaweed kelp, hydrogen peroxide, salts of phosphoric acid, and then sex pheromones on insect side of things. Those are the natural substances that could be classified under biopesticides. And then we have the microbes. So there's a list of assortment of microbes. I did my PhD work actually on Coniatherium in white mold. So that's one that attacks, and we'll talk a little bit about that. But then there's an assortment of bacteria and other things. And then my note on the microbes is, if you think about, we already have a hard time sometimes when I want to do a disease evaluation, getting the disease to establish in that field. The microbes also, when we talk about the pest disease triangle, the environment's going to really influence their activity. So it's important, if we're going to introduce something that's living, that we introduce it in an environment that encourages it to work on the disease of target. And so then there's also plant-incorporated protectants that may induce plant resistance, and Bt gene would be one, but that's not organically available. And then the idea is, why are we using biopesticides? Because they're generally less toxic than our conventional products. They only generally target that one pest that we're trying to manage. They may be effective in small quantities, and generally those products have no reentry interval or pre-harvest interval as well. And so if we look at how are they working in our pathogen systems, there's generally 5 modes of action. Antibiosis, so that's actually a microbial, it's going to kill that pest. Maybe it's parasitism and predation, so that, like Coniatherium, it's going to parasitize the inoculum in the soil and break up the sclerotia. Maybe they're competing for leaf surface, so they're on the leaf surface and they're outcompeting that spore that may land from the pest. They may also put out biochemicals that may inhibit the disease from developing or turn on the plant's natural defenses. So that's kind of how those biologicals work. They may have 2 or 3 modes of action. By having this multiple modes of action, there's less chance of pesticide resistance, and that's the other side we think about when we're using some of the conventional fungicides. And so again, I mentioned there's this Crop Protection Network has this new biopesticide book that's available. It has a list of all sorts of biologicals out there. For example, I got Actinovate. It kind of talks about the targeted organisms, the crops that it's labeled on. Just a tool to look at if you're trying to target, are there some products that are available that I might be able to put in my system if I'm having a problem with a specific disease. And then again, here's Coniotherium as well, so a mycoparasite. And so do they work is a big question, and that's what led us to this project. Some of them work well against some pathogens, but not others. These biopesticides are not curative. So that's the other thing to think about is maybe it's a stepwise. We get a little bit of control, but not maybe what we see when we use a fungicide. Environment is going to be the limiting factor. Are they being put in conditions that are conducive for that biopesticide to work, or is it working against that product? And then look at, has there an interaction with a biopesticide with a crop or cultivar? And then when do we need to be putting them out to prevent that pest? So again, these are some of the questions we need to ask if we're looking at some of these newer products as they come online. And so as we move into managing diseases in organic crops, even in conventional crops, there are 3 things that got to occur. We first have to have that susceptible host, whether it's a corn species or soybean. Maybe we've selected a variety that's more resistant, and understand where is that susceptibility, where is the pathogen getting in. So for example, SDS, Fusarium, they're getting in early at the seedling stage and infecting the root then. We may not see the symptoms of sudden death syndrome until the reproductive stages. Again, it's too late. If we look at our foliar diseases, they're when we're have those full leaves open for infection. That's the susceptible side of the growth stage of the host. Then where is the pathogen? Is it already present in our fields? Is it overwintering? So gray leaf spot, now tar spot, white mold, they all overwinter in those fields. So they're already present in that field. Some of our diseases aren't here. So southern corn rust, I don't know if you worry about as much here in Wisconsin, but we do monitor when those spores move up. If they come into central Indiana in July, then we may have a problem with this disease that moves in every year. But every year, we clean the plate, and we don't have to worry about it until the spores move in next year. It's not there year after year. So understanding where the disease are, and are they present in the debris? And then finally, what is the environment that interaction requires? Each pathogen and host has a specific environmental conditions it requires for that spore to germinate and to get into the plant. And so if the temperature and moisture and leaf wetness or whatever the requirements are unfavorable, then our risk is low. And I wish I had a crystal ball to predict what's going to happen this season so you know what you need to do, right? We have new tools that are available that we're trying to predict when the disease risks are high, and I'm going to share some of those. But in years where we have the favorable conditions, even if we do everything right, that disease may push through and still cause problems. And so that's important to understand as well. So again, 3 things that need to occur for a disease to happen in a field. And so with this project, we decided to target 3 diseases of concern in Indiana and Michigan and Wisconsin, picking our 3 grain crops, so corn, soybean, and wheat. And what we targeted was, we went after Fusarium head scab for wheat, because we know that's a concern, white mold in soybean, and tar spot in corn. So those are the 3 diseases we started with to try to look at this project moving forward. And so we went and looked at what products are out there and labeled, maybe what products are working in other systems, and tried to identify what could be used in each cropping system. So we had some mineral-based with coppers. Coppers are a protectant. They need to be there on that plant surface. They probably are only there 7 to 10 days. So the question is, do we provide enough protection if we put a copper in versus something else? So we looked at some of the coppers. We looked at some of our botanical pesticides, so paste setter, which has got a giant knotweed extract that acts in eliciting plant host defense responses. We also looked at an oxidation reaction-based product, Oxidate. This product is designed almost like a cleaning solution. When it's applied, it's going to kill all the spores it comes in contact with, and then probably is only there for 4 hours. So you got to think about, if we're going to put that out, do we have something else to then come in and protect the plant if we've cleaned the surfaces of the spores that have come in? And then we have some of those living microbes. So we had actinovate with streptomyces, Botrys-stop leucladium, double nickel bacillus. There's a number of double bacillus species, but we'll try to look at some of those depending on the crop that got inserted into these trials. And so first, I'm going to talk about the Fusarium head blight. And so this disease survives in our crop debris. It survives, it also infects corn and wheat. So if you think about it, we have those rotations. If we go from corn to wheat, we've built up a little bit of issue. If I look at this season, actually corn had a bigger problem at the end of my season in Indiana with Fusarium head blight and inflection, or Fusarium getting into the grain and causing mycotoxin issues. So it's important to understand where it's surviving. So it's in the debris, and then preceding a wind and rain, those spores are released and they land. And in wheat, our susceptible stage that we're targeting is this flowering head. That's where they get in and cause disease. And so if we think about how are we going to manage this disease, I forget if I got this animated, so we want to protect the flowering head when it's susceptible. And so if that's what we're thinking about, if we know we'll reduce our risk for Fusarium head blight, that's when the protection needs to go out. And then, as it develops, this is the bleached head that you might see. And that'll lead to these shriveled, chalky kernels. Some of those kernels, the Fusarium produces the DON, the vomitoxin that can cause issues in our grain. And so again, a lot of work has been done on best timing for using a protectant to protect these heads. So this early 10-5-1 is the optimum timing for protecting wheat heads. And so a part of the work that's been done on trying to figure out Fusarium head blight is we developed this risk tool. So it's pulled in the weather data that says, yes, the weather data is conducive for the disease. You can also say that I have a susceptible variety or I have a moderately tolerant variety. And you can use the system to say, has the disease, is it turned on? Have we had those environmental conditions that's going to really cause disease when we're at flowering? So here's an example of what 2023 looked like. So Indiana, generally, we were pretty low risk. It wasn't red, only some small pockets. If I changed the map and went into a moderately resistant variety, we clear, that risk. So that's the first thing, again, and I'm going to probably keep pushing this. If we can find resistance for these diseases of target, that's our best option in trying to manage disease. So again, at low risk, Indiana, I hardly saw any Fusarium in our plots. Even where we inoculated, I got 1 or 2 heads of everything we looked at. So the disease was relatively low. But this could be used to try to predict if we go red in that time frame when we're flowering, maybe we need to think about when we make an informed decision that we may need to put out a protectant for that disease. And so when we looked at this in our trials that we ran for the last 3 years here in Wisconsin and Indiana, we picked 2 cultivars, one with a little more susceptibility and one that had some resistance. And then we had 5 products. We did compare to our standard fungicide. And then we looked at the champion as our copper, pace setter, sonata, and actinovate as options for controlling Fusarium head blight. And so if I look at the combined data for Indiana, and I think I broke them apart by each state because we had different disease pressures in each location. So the summary of those 3 years, here you have Harpoon, my blue, so our resistant line, Kasiaski, the orange. So you can see index is the amount of incidence versus severity of the disease, and then severity is just how severe was that head of that wheat infected with Fusarium head blight. So you can see, first of all, the Harpoon, we can significantly reduce that incidence and severity if we plant something with resistance. And this is going to be my harping for organic disease management, identifying those things that have resistance. Whereas you can see we had higher severity in that susceptible cultivar. If we look at Wisconsin, similar results. Again, the Harpoon had low index and low severity, whereas Kasiaski, we did see a higher infection rate, so a higher disease development in that susceptible. If we look at the products, and so this is in Indiana across those 3 years, and each of these slides I tried to show where my non-treated block is. So it's the one that's got a design in it compared to the other products. So again, Prosario is my fungicide. So as expected, our fungicide did provide protection, but you can see here, for instance, the champion actually was similar to our fungicide, and then we had some that we did not see any protection on those diseases. They weren't different from the non-treated control. Over here, if we look at incidence, again, the Sonata had less heads with disease, whereas this was I'm trying to think, what do we had here? Oh, and okay, difference in hybrids. So you can again see lower disease in our resistant hybrid versus our susceptible hybrid. In our susceptible hybrid, the Sonata trended to reducing disease. I would say some of these more biological products, we may not get that statistical separation, but you can see we're trending over 3 years where that one provided a little bit more protection. If we look at Wisconsin, they were able to merge it across each of the hybrids, so they had the same reaction. Generally, only the fungicide worked. So again, if we're moving forward to wheat, again, I want to target, let's try to find those hybrids, those cultivars that have resistance, and start with that first, and then we may not need to rely on additional inputs. If we look at the DON, so that's the important part, how much vomitoxin was isolated from those trials, you can see Harpoon had less, again, less infection, less DON developing versus the Kasiaska. And then also Fusarium damaged kernels. Again, we had less in damaged kernels in that resistant line versus the susceptible. If we look at the fungicides or the biologicals, again, Sonata was similar to Prosario, but Champion actually was one that had less disease develop, less damaged kernels develop as compared to the non-treated control in Indiana. If we look at what happened in Wisconsin, really they didn't see any effect of those treatments again on minimizing DON or FDK, but again, starting with resistance and versus susceptibility is our best option for managing that. So you can see the Harpoon had significantly less mycotoxin as compared to Kasiaska. So again, emphasizing the importance of trying to find those varieties with resistance for Fusarium head blight. If we look at the yields, generally, the yields were flat for treatments for Indiana. Our Harpoon actually out-yielded the Kasiaska, so that's good news too. Actually, a cultivar that has good disease resistance and good yield potential. I like that. Sometimes we don't see that in other diseases. But again, that's from 3 years, again, if we could identify that variety to use. So for Fusarium head blight, I think variety selection is our goal. We got to find that high quality seed with resistance if we're worried about that disease. We may think about managing our residue. Are we rotating from corn right into wheat? Is that going to increase that risk because we have that residue available from previous seasons? Maybe putting a legume or oil seed, which are not hosts to Fusarium, in that system. And then to think about where are we managing our irrigation. If we're going to irrigate, we want to avoid that during flowering, where there is a higher risk with the disease getting in at that point. We know there's a number of conventional options available at Feeks, but again, some of our organic options are still limited if we want to put out a fungicide application. And then also to note, as we get ready to harvest this wheat, it's important to go look at it and see if you do see that blight in those wheat heads, because you could increase your fan speed and other things to push out some of those lighter damaged kernels so you don't have that in the grain when moving forward when it's getting ready to sell. So that's my quick overview of Fusarium head blight. Any questions before I move on to the next disease? Yep. We kind of asked questions around and identified varieties of seeds that were available to get those that we used. Yep. Corn? Yes. I can, yeah, no, we'll stay on Fusarium because I'm going to switch gears and go to a different pathogen. So I know from this season, and I haven't had, since the 5 years I've been in Indiana, we haven't had issues with vomitoxin in corn like it's ramped up in the last 2 years. And I know the calls I've been getting all this winter are about high mycotoxins in our grain corn. So thinking about that, if we talk about the disease cycle, just like with Fusarium head blight, we have years that are bad, and then we go lay low, and we may build up inoculum, and then we have the right environmental conditions. And so if I look at corn, where we need to be protecting, if we're going to worry about that, is the green silk stage is where the Fusarium is getting into the corn ears to cause the problem. So that's the stage. If we were looking at flowering here in wheat, green silk at corn would be the protectant stage if we were going to target a fungicide then. But then it's also important we were rotating, we probably were building up inoculum, and then we had the perfect storm of conditions for this season to have that infection.
So that's what we're trying to figure out, was just the first 2 weeks of August was hot and humid in central Illinois.
Right, probably. Yeah, that's most likely what triggered it. We had the optimum conditions for the infection when we were having silks out. And if I think about our season, I had growth stages all over the board trying to determine when we were going to spray different treatments. So I think there's another thing is we had various growth stages at different times. So there was green silk coming out across a longer period. So we probably opened the door for more infection to get in when the rains came back in that region. So that weather conditions is what really kicked that new disease, or in the corn.
The other part of the question is, thought, what you say is, we're using rolled rye ahead of soybeans, we're using cover crops after the wheat ahead of corn. Are we just getting so much inoculum out there that it would?
Yeah, so the cover crop interplay is a big question that we haven't answered on how that's providing bridges for these these pathogens to keep persisting. So that would be the one thing, but that rotation from wheat into corn or corn into wheat, we're building up the inoculum. Even if it was a low level, they still have infection probably going on that we're maintaining those diseases as they move along until we get an outbreak. And so now after this season, I suspect our levels of fusarium are pretty high in those fields. So if we've planted wheat in those fields, I'd be keeping a close eye on that scab tool initiative to see for wheat if we have the moisture conditions that are conducive, because I would say this season the wheat didn't lead to the corn that, usually soybeans are following our wheat anyways, but I think we were building up year after year with these rotations where we have similar hosts. And then moving forward as we talk about some of the forecasting that we're doing in the other two diseases, we are doing some spore trapping. And so that's on our list of trapping these spores and trying to build them into if we see the spore release early and we have the conducive conditions, can we pre-warn that? But we're still building all that data up. But that's where I think, and I've been pushing in Indiana, we lost our pathologist retired and we didn't fill that position. So I've been saying we need to continue to expand on that data for grain mycotoxin evaluations.
Is there an organic option for fungicide in the corn?
I don't know. I didn't look at it, so I won't have an answer on that one. So that would be the question of where do we take it the next step.
So
Yep.
how much does tillage affect the fusarium for wheat following corn? You want that
So
ground as well?
tillage is going to bury that debris,
Yes.
and then that won't be readily available on that surface. And each disease is going to depend. The fusarium's there probably, and we've buried the debris, so we've eliminated that top layer. I'm not going to tell you to till because you have your other soil health benefits of tillage. So that's the other problem when we talk about tar spot and other things, is tillage may not help us enough. The inoculum may already be there to cause the problem in the next season. But the rotation, so if we can get away from these rotations, a longer rotation, that may help limit the amount of disease that goes in. But I think you need to think about as we insert cover crops, are those cover crops, are they host to the fusarium species? So are we continually building them up over time too? And those are lots of questions we haven't answered of that. There's some work being done on some of the crops, but again, that's.
The question I had with another farmer, we were talking about this, is trying to no-till the wheat after corn. Is that an option you could try, or is that not recommended?
No-till the wheat after corn? I get, but you're going to leave the corn. I guess the debris is going to be really readily there, available, and it depends on how well you can get the wheat in. I guess there's other things that I would look at is how well you can get the wheat in the ground following the corn. But corn to wheat, that's where if this fusarium was there, it's in the stubbles, it's in the stalks. So it doesn't only just go to the ears. We can have a fusarium stalk route. The same pathogen gets in that whole plant, so it may be present.
The breakdown of the cover crop of all the weeds when it's in the soil or stay on the soil. That's the breakdown for how the soil will capture the different
Right.
spore, or the degradation is higher when
Well, so, and I don't think we have all the answers on how we degrade fusarium, per se, in the soil, because it's actually grows very saprophytically all over, and it just finds that niche once in a while with the corn, but it's still pretty happy on debris there. So, and each disease is going to be different. So when we get into white mold, I'm going to talk about how roller crimped rye may help provide some other barriers. But again, it gets back to understanding the disease cycle and how our different rotations may influence different diseases. And so, and there's not a perfect answer. That's the problem, is because we may target one disease and we get that to go away, and then something else is going to find the niche. So I wish I had a one-size-fits-all answer. One more question, and then I'll move on, so I don't run out of time.
You are planting wheat after corn. Are those years higher than corn? You have 3 years.
And I have no idea, so we haven't done that. So that's some of that. The alfalfa should have helped knock back some of it because it didn't have a host. But did you manage your weeds or other things that could have? That would be the other question in that system as well. I'm not going to get into the weed management side of those as well. But we got to think about the hosts. So if fusarium has a wider range of hosts than just corn or soybeans, and so. But then I switch gears here, and so I think I got tar spot next. Very specific disease for a specific crop, and so that may help us try to manage it better. So this is tar spot of corn. How many of you have seen it on your farm here in Wisconsin? Right, have you seen it at this level? Yes, okay. My talk, and so usually I spend all my talks talking about just tar spot management, so this is a little change, talking a little bit about organic management. I just want to note though in Indiana and from our data, that if you go out in the field and your ear leaf and above is this level, this 5% or more stromata, it's game over. Don't even try. If you spray a fungicide, you're not going to stop it. We're wasting our product. It's already gained access. When we have a favorable season, I can go from the leaf on the left to the leaf on the right in 2 weeks. And depending where we are, so if we're at dough and we lose all those leaves like the middle, that's where we're knocking the socks off our yield, okay? So we got to be ahead of the game, and I don't have all this data, but you can be out scouting. When I'm at 1%, so 1 or 2 stromata on a leaf, I probably can find it in every plant, and you still have time to do something there conventionally. So that's just a note on, we got to be ahead of this one to try to control it. Problem is, if you hadn't seen it on your farm, this is when you're going to see it and you're going to call us, and it's too late. So a little bit of what we've learned on the disease cycle. The fungus is overwintering in our residue as those stromata, and we're going to talk about sclerotia in a little bit, but the stromata have this melanized structure just like our sclerotia that allows it to overwinter. Think about the billions of stromata that we're putting back into the field each year. And so we know it survives in the stromata. We've taken samples in the spring and we're able to get germinating spores. This pathogen I can't grow in the lab, so we're having a harder time trying to understand the biology because I can't grow it on a Petri plate. I can't grow it like fusarium. But we do know we can get living spores. I suspect from the fields where we first started with this disease that it's overwintering more than 1 year. I can go into, there were times back in 2017 where I suspect the corn was infected. We didn't notice it. 2019 corn, epicenter where that disease started, and they were previously soybean full tillage, so it was clean bed. So I suspect these stromata are surviving even when we can't see them, if there's not even visible leaf tissue there. They're there. So the stromata, they release their ascospores, they forcibly eject them up into the lower canopy. So if you've had a history of disease, we have the right conditions, those spores move into those lower leaves. You can still scout and find them in those fields in the lower leaves as they continue to move up. Usually takes us about 14 to 21 days for us to see those first stromata after that spore drop. And then the cycle is going to continue. And so we're still trying to figure out how fast that secondary cycle goes. But if this starts in early July, I have a long period to get to harvest in September, October, multiple cycles that can occur, and that's where it ramps up. And so we've been trying to work on this disease. And so just as a background, this is the movement across the United States. Every time I turn on county yellow, that's where a positive county turned on from 2015. So it was first found in Indiana, Illinois in 2015, and it continues to spread. So this is where we talk about management. We have these spores all releasing. And once they get going, the spores are throughout the whole Midwest. So this is where rotation and tillage may not play a big role in trying to manage this disease as well. So you can see it continues to spread. We have a pocket that started in Florida, and probably that's the pocket that's moved into Georgia, but you can see kind of it continues to spread, and I don't have the final map there. I can't stop it. So again, new counties every year. It's going to depend on your weather conditions whether it's going to be a problem in that season. So for our organic work, what we tried to do is identify some hybrids. So in 2021, we started just a single hybrid, and then our counterpart in Wisconsin, they found a resistant one, so we put that into the trials in 22 and 23, looking at 2 hybrids. And then we looked at some of the fungicides that may be available. We had 4 products that were labeled for corn. We compared to our conventional control, And then we tried to do different timings. So in Indiana, we went under standard VT tassel application timing. Wisconsin had used tar spotter more readily, so they used their tar spotter to trigger applications at V12 and R2 in 21, V8 and R1 in 22, and then V10 in 23 is when they applied that. If we look at the products we had, so our standard, we went out with Headline Amp, that showed to be pretty good against tar spot. We looked at Serafel. I had used that previously in other crops. Looking at that one, if we could see it, was a Bacillus amblyofaciens, a bacteria, Streptomyces, actinovate. We put a copper in there, so copper badge, and then we also looked at the oxidate. And we just looked at the one product, okay? I would say if I was gonna really think about a program, the oxidate is, again, the one that I said has a 4-hour window, it's killing the spores. I would think about maybe probably adding that to something else, a protectant, so maybe the copper. Moving forward, that may be some option to do where we knock down the spores that are on the leaves, and then we put a protectant out to protect against the disease. So if I look at, here we have the separated Wisconsin and Indiana, the disease that developed throughout the season. Wisconsin, generally only their fungicide worked. Headline Amp worked against reducing tar spot. In Indiana, we actually saw in 2021, which I would say Indiana, that was my really bad year. We had a severe epidemic in 21. Actually, that copper held on, so it was providing some protection for tar spot. And then even the oxidate, actinovate, and Serafel did reduce disease as compared to the untreated. So there may be some other options to add for managing tar spot based on some of our Indiana data under that season. But if we look at the yields, generally our yields were flat, but we do see that maybe actinovate and oxidate were trending to increase yields over the non-treated. Not quite different from our fungicide yield. So again, maybe there's some products that are protecting yield. That's the ultimate goal. Again, in Wisconsin, really the only one that worked was Headline Amp, the fungicide. And then in 23, when we added the hybrid, so in Indiana, we had high disease pressure and our hybrids performed about the same in that field. And then looking at the fungicides again, the copper in the second year is showing some control, maybe not quite different from the fungicide, but better control than the non-treated, even though they're statistically not significant. But again, we're trending for a little better protection. And I think if we look at some of these organic OMRI products, we may have to, they may not provide the silver bullet like our fungicides, but they do provide maybe a little more protection than what we would have gotten had we not treated at all. And then if we look at Wisconsin, they actually saw a separation between their hybrids. So the 96 did significantly reduce tar spot. And in some of our trials where we've looked at other hybrids, I do see a significant effect of finding a hybrid with some tolerance with resistance to tar spot. We can reduce the disease and keep it about 5% if we can identify those. So that's the thing we need to push. The biggest issue we're having is generally, if we don't have disease pressure, they're not the higher yielding hybrid. But if you're worried about this risk, if we can initially start with resistance again, we reduce that initial risk of that disease in the field. You can see there's a trend still here, but they did not statistically separate out for the fungicide treatments. In Wisconsin, I guess Serafel is closer to Headline Amp than some of the others. And then in general, our yields didn't separate for that year. So yields were flat. Red is Wisconsin, gold is Indiana. You can see they really didn't separate. But the idea is, can we minimize some of that disease pressure in a year where we have significant disease? I would say for 23 in Indiana, I saw about 20 bushel protection with some of our fungicides. So we did have some yield losses where I think Wisconsin didn't have as high pressure this year in some of their plots. And I think that's all. So we are trying to expand this because that's just 2 locations in multiple years. And so we've added other locations that are trying to help evaluate some of these products. And so across 5 locations, we pulled some of these efficacy data to look at. So you can see badge is one that's trending, the copper trending to protect against not only tar spot, but maybe some other diseases that are out there. So that would be gray leaf spot or northern corn leaf blight, depending on the location. So we're trying to build this data. We have really good efficacy data for the conventional products. And so what we're trying to do is build better information, better efficacy data for some of these OMRI products as well for you to utilize. So that's some results we saw last year. But again, I think most of the disease pressure across the region was flat, so no impact of fungicides. And I would say generally in Indiana, we didn't need to spray fungicides except for maybe where there are pockets of tar spot this year. And so again, back to using resistance. If we can find resistance, use that. Fungicide application can increase our protection. I didn't put it in here, but using the Tar Spotter app is going to be critical. I think it's my next slide. And then impact is going to differ year to year. Again, on those environmental conditions, once the disease is there, when is it going to really ramp up in that canopy? So again, for tar spot, you have first understand is in your area. So I think most of Wisconsin's filled in. All of Indiana, I think we have 4 counties that I haven't had a sample come in to confirm it. I suspect it's across the whole state. Using this map now, I tell my growers that right now, if you went and looked at the map, it's all gray. So we're indicating where we have found it. And then when I turn it on, we're out early trying to find those first vines. It's telling you to get out and scout. I'm not saying get the airplanes to spray. I'm saying get out and scout. Can you find it on your farm? We found it active this season, and see where it goes. So again, now those maps are available. We have the Tar Spotter app and the Field Profit app out of Wisconsin that are helping predict the weather conditions that are conducive for tar spot. This year it's finally really working in Indiana. We've had enough weather data to pull it in. And this year it would have triggered a spray at R2 and R4, so later spray, because the disease didn't really start till August. We first found it in July, but then we stayed hot and dry and we stopped the disease. And then we had rainfall in August that triggered it back on again. So there's some tools that are available to predict. For conventional products, there are the efficacy charts. We're just working on updating that. For this season, I hope in the near future we get enough data to start putting efficacy charts out for some of these OMRI products as well in grain crops. So that's our goal. And I'm just gonna say, get tolerance, resistance out there. Leave check strips if you do something new. It's important to understand what you do, get you a return on investment. Did we protect something? And then just understand what is the risk, what diseases are you worried about? Maybe it's not tar spot, maybe it's fusarium. So it's important to understand what disease we're targeting. Any questions on that? I don't know how much time do I have. All right, I gotta turn to any questions on corn before I switch to soybeans? Yes.
So if you've got a resistant variety, has it been long enough testing to know, so are you just selecting for fungus that can live on the resistant variety, and then the next, if you planted corn on corn, would the spores that survive be way more likely to be able to infect the variety the next time?
So for tar spot, our resistance is not true resistance. So it depends on your type of resistance. So if you have a true resistance, it's like a yes or no answer. And if we plant them over and over again, then we're gonna select for the population out there that has overcome that resistance. So that's kind of what we're seeing if I talk about soybean cyst nematode. We put the same resistance packet into most of our soybeans. Now we've grown them for 10 years and we're starting to see the populations shift where they're overcoming that. Most of the resistance that we're looking at right now in corn is a graduated resistance, so there's not true resistance. Something is slowing it down with that plant-host interaction. We really are still trying to figure out what lines are resistant. We just used some of our data to identify some that had less disease compared to others that had severe disease. So right now, I would think rotating is not, we're not going to select for a resistant population yet because I don't think our resistance is one-on-one interaction with the pathogen. It's a more broad spectrum resistance. Just like if you think about our fungicides, if we have a site-specific action we select over time, we're going to select that because it could just make one modification versus a broad spectrum. We're hitting multiple areas within that pathogen, less likely to develop resistance.
And it's a direct, like you could count the number of lesions on a leaf and directly correlate that to yield at the end in those varieties?
We're still not there yet. So it depends where you start. And so I can actually see, so some of the lines that I said had that I'm identifying as moderately resistant, they look good for a long time, and then something happens and the plant's starting to shut down, and as long as there's green tissue, this disease is gonna find it and get into it. So I can see where all of a sudden the disease takes off in some of those hybrids after it maintained at a low level. So the hope is we got it, the protection of that yield, and then, see that. So that's where some of the ratings and I know the industry's having a hard time rate them because it depends when they rate. And if they wait too long, even with our fungicides, this disease pushes the limitations of all our fungicides. It tells you how long they're there, how long they're active, how much area you covered. I can see where the lower leaves don't get any coverage and they're just eaten up. And so that's the other side of this, is this one disease is gonna push us. And it really, those years where we're like gonna have a bumper crop, it's probably a year we've had really conducive conditions for this disease. Unfortunately, we're still early on in trying to figure that one out. But yep, I'm hoping 20 years I can say we have resistance and we don't have to worry about fungicides. Yeah, in the back.
Is anyone looking at soil health characteristics and whether or not that makes the plant resistant, less resistant, or more resistant?
Not directly. We do have some trials where we've looked at reduced tillage versus no full tillage on starting the epidemic. What we're seeing is there's a delay, so we can delay the onset or reduce the initial disease, but then once we're going again in season, it blows up. And that's the problem, is it's because those spores are moving, you may do everything right to try to minimize disease in this field, and we may have a spore drop from the next county over. Not even the neighbors. It may be just coming from Indiana, maybe taking it to Illinois, and Illinois, Indiana, you know what I mean? It may be crossing over where we have, and then the environmental conditions just continue. The environmental really will push when this disease and how fast it takes off. And so that initial picture, I mean, usually every year, for the first 3 years of dealing with this, I had great trials because I saw that every year. And then 22, we had a dry year and I didn't hardly have any disease. So we need that data to pool into our apps and stuff to help predict what we're trying to do. But we're still working on it. Fusarium has been around since the early 1900s. We've been fighting it and we still haven't found the answers for that. We're like 6 years into research for tar spot. So that's what I would the changes that we know on different pathogens. We're trying to learn as fast as we can. So these apps are really useful, I would say. So if you see me turn the map on, I would pull up your app and start running the app to see if I had conditions that are conducive in my fields, and I need to keep an eye on that for when the disease is active. You guys just keep me busy, and I got 3 other new diseases in Indiana to deal with, but we won't talk about those today. And so then I'm gonna switch a little bit. So white mold, if I could pick, this is the pathogen I would pick, just like Damon Smith likes to work on this pathogen. I worked on this in peanuts back in the day. This is probably the worst season we've had in Indiana since I started back in 2018. This is, of course, not where I have field trials. Of course, it's the next field over where we have a white mold outbreak. But many of you that have seen it, we have this crooking of the plants, and if you open the canopy, we have that severe white mold infection. Looking at the disease cycle and how do we manage white mold if this is a problem. The biggest issue with white mold is once it's on your farm, it's probably going to be a problem in those areas for a long time because these sclerotia can live in the soil up to 10 years or more. And if we think about this last season where I had areas that were hot, we just reintroduced more sclerotia to that field for the future season. So again, we have the sclerotia and the debris. They germinate, they produce these little mushroom-like structures called apothecia. The apothecia release their ascospores. The infection occurs at the flowering stage, or when we have necrotic tissue, that's when the ascospores are getting in. This is a little different from when I worked in peanuts. Actually, the sclerotia could directly penetrate and get into the peanut stems that way. Whereas in soybeans, we know they're getting access through the dying flowers. So think about where is your flowering period? Did we have a longer flowering period this season with the conditions we had? And so in Indiana, that's what led to, we had a wider window of when infection could occur. And then once it gets access, it'll develop these lesions on the stem, produce more sclerotia, and it's shed to the soil. So compared to tar spot, really it's one cycle of disease. We're not only having one spore release that gains access, and when do we need to protect the canopy for that access? Whereas tar spot had multiple cycles occurring. So in this trial, we looked at a couple things. Back when I was in New York, there was some work done on the roller crimped rye, protecting snap beans from white mold. So we did a tilled rye cover crop, and then we tilled part and then roller crimped the other part. We identified 2 cultivars. So Wisconsin had this MN14 and 5282 resistant to white mold, and then a susceptible Duane and Dwight, and then foliar fungicides. So we looked at 4 OMRI products compared to a conventional and non-treated control. In Indiana, we went at R2, so full flower stage. Wisconsin did R3 and then sporecaster applications. So again, the products, similar products, Actinovate, Botry Stop, Double Nickel, Serafel, ones that were labeled for soybean and white mold. And then we used our conventional product. Wisconsin used Endura, and I used Approach But generally, we're throwing the conventional in there just to give you an idea of what it compares when we look at the standard conventional products. So of course, none of this is done on organic land. And we also, in Indiana, we inoculate our plots. So we had small plots. We did a split plot, so the tillage was across the plots. Cultivar, 2 treatment subplots, and fungicide 6 treatments. Wisconsin has a nice field site that has a history of disease. We're continuing to build ours in Indiana, so we added inoculum. And we added our inoculum before we roller crimp the rye. So the idea is we put down more sclerotia, then roller crimp the rye over the top of it. And then we collected disease ratings and yield. And so if I look at Indiana, so we had differences in disease development. So again, Indiana separate from Wisconsin in these trials. What we could see is the MN has resistance, so it remained really low. We couldn't find any disease. But you can see in the susceptible line, we could significantly reduce disease by that roller crimped rye. And so if we go back to that disease cycle, what that rye is doing is we're laying the rye over those sclerotia, and it's inhibiting the proper formation of those mushrooms from developing. So we're stopping the mushrooms from developing possibly, and then maybe even actually capturing those spores. So we're providing a physical barrier for the disease to get in the canopy. And then yields on those, really we didn't see any difference in yield between roller crimped rye and some of the varieties. I know there were some years where we did see there was differences where one variety did better in the roller crimp than the other. If we look at, again, this is looking at the products. Generally, we didn't see any differences in our products. There was no significant differences there since our disease incidence was low, and then resulting yields are flat when we don't have the disease pressure. If we look at what happened in Wisconsin, what they did see is a change in stand count. So the tilled, actually the roller crimped rye had higher stand count than the tilled, and then there were some differences between the different cultivars and the stand count. They also saw that, that Minnesota 1410 had less disease develop, and then higher yields as compared to the susceptible. So again, we're back to the whole identify the varieties and cultivars you want to use if we're targeting a specific disease. That is the first line of defense for these diseases. And then if we look at the tillage method, actually on disease incidence, the roller crimped actually had a little higher disease in Indiana this last year than our tilled, but every other year the tilled had less disease. If we look at what happened in Wisconsin in 22, did I go backwards? I think I did, yes, I did. So then if we look at Indiana on yields, again, no difference in tilled versus roller crimped, no differences in the varieties, and no differences in that fungicide users or the products we tested. Again, if I don't have disease pressure, then we don't have differences in yields in what we use, and so we should save them and only apply them when necessary. If we look at what happened in Wisconsin with our disease, again, in this last year, again, that variety, less amount of disease. They did see some differences in fungicide, the treatments, and the Endura, the standard one used in our conventional system, provided the best control, but there was some slight tendency of that Serafel to work as well. So again, these are the kind of the data we're trying to pool to give us efficacy ratings for using some of these additional products. If we look at yields by variety, again, that resistant line out-yielded the susceptible, and then in their tilled, they had higher yield that year. So again, the roller crimped rye may influence how well that variety yields, depending on the disease. And then we, again, are expanding, looking at, in soybeans, again, some of these products. So here I have black is frog eye leaf spot, green is white mold, and you can see generally still we're seeing that we're not seeing any influence of these on some of those other diseases as well when they're applied across the region. But this is the data we need to try to keep pooling to say yes or no they're going to be effective for the various diseases. And then again, yields were flat relatively for that. And so if I think about that disease cycle again and where we're trying to minimize the risk, so we can look at our culture and agronomic practices, row spacing, population, resistance, and we're acting on that side, the host side of the disease triangle, and maybe trying to influence that microenvironment with row spacing and population. So that's how we're trying to act and break that disease cycle. If we looked at this roller crimped rye, it's going in here, inhibiting some of that sclerotia germination, those spores, the ascospore release. If we look at our biologicals and fungicides, they're going out foliar. We need to protect where it's gaining access into the flowers. And then finally, there's biological out there, Coniatherium. It is a mycoparasite of the sclerotia, and the idea is we're going to go after that initial inoculum. So I'm thinking it's a layered effect, a long-term evaluation of how we try to reduce disease. They may not provide disease control for one season, but if we can break down this inoculum availability in the field over the long term, maybe we reduce the risk for it to happen, or outbreaks to happen in the future. If we protect with fungicides or biologicals here, then we're not returning some of that sclerotia back to the field. And again, it's kind of like a weed seed bank kind of assessment. We want to reduce the inoculum bank in that field where we have problems. So that's kind of where I would layer the different management tools for white mold if I had problems in my farm. Yep.
Is there a relationship between growing degree days for the release of the spores in the blossoming of the
there's been a lot of work done, and Damon's group has done a lot of work on flowering and when the spores, when those mushrooms are released, and it's like they've evolved together. So the pathogens identified where the canopy is the best time to gain access to cause the disease. There's been a lot of work in the Wisconsin group. I didn't have that data. I've seen some of the data. We get on their hands and knees and scout these fields for when these mushrooms are appearing, capture the spores, and then relate it back to the weather conditions. And then they can relate it back also to the host conditions. So what stage are we in the soybean? Is the canopy closed? And so if you look at that, I don't know when I put the Sporecaster app, but it's looking at when is the canopy closed? When is that microenvironment building up to minimize? And so when can we target, when we got to do a targeted fungicide application for that? So there's more data on this aspect than some of the other diseases we talked about today.
Can you speak to, shifting gears, speak to white mold and sunflowers?
I haven't done any work on white mold and sunflowers. I use my sunflowers to build my white mold plots. So it's going to be the same, so the white mold, the problem with white mold and rotation is it's got over 500 plant species that it goes to: alfalfa, tomatoes, sunflowers, cucurbits. This pathogen's got a broad host range. So if we have a pocket of it, you don't want to move that pocket to another field because it's long-term survival and then a wide host range. So it even has weed species that are, many dicots are susceptible to white mold. I don't know if I answered the question. So there is some work out of North Dakota. So I guess some of the people in North Dakota may be doing some white mold work. And I'm not quite sure. We inject our sunflower heads to try to build up white mold inoculum and bury them in the field. So I know, white mold is one concern for sunflower because it is very susceptible. But I've seen it eat a cabbage head, right? It can get into cabbage. It can eat up a tomato plant. So as a pathologist, it's a pretty disease, the way it just attacks a plant, but it's not good for you farmers. And so if we look at some of the work, really residue management may be helpful. This roller crimped rye may show ways to impact that spore release. If we can, again, resistance. If we can get them out there. White mold, it's not a true resistance, so they have a moderate resistance available. And then this fungicide flowering, if we can. And then, we're doing some more work on biologicals. I didn't have time to present all that data, but we're moving forward or trying to figure out where can we incorporate this. Is it better to put it in the fall versus the spring to go after targeting those sclerotia? Again, it's targeting. Can we reduce that initial inoculum to minimize the risk in season? And so there's all these tools I mentioned, the Sporecaster app and the Tar Spotter app. They're all available out of Wisconsin. There's also the Field Prophet, which is available, which is actually pulling weather data to predict out 7 days beyond that. So those are available. And then the Biopesticides is a new book that's available online. You can't print it, but it's an online tool that's available where we're trying to pull some of that new information in as well. And then, none of these they're not curative. We just have to use them in a preventive manner. We need to integrate them, layer them in. There's no silver bullets. So we need to think of how they integrate with other pesticide strategies, or, if we put out a living biological and it's a bacteria, will adding a copper at the same time attack that bacteria. So we need to think about how we integrate with other pesticides, and then understand that we need to know, first of all, what we're targeting. And each management task may be different depending on that target. And then for sustainable disease management, even when I talk to my conventional growers, host resistance when available, understand your disease cycle, where do we need to do things if we're changing practices. It's important to understand what we're doing, leave test strips to see if really something new really works. And I'm gonna note on this new technology, it is important to understand I have a disease that's gonna push the limitations of our fungicides and our application equipment. So this is an example. This was not intentionally strip test of tar spot. The drone coverage really was only effective in the 15 feet underneath that drone, not the 30-foot width that they thought they had. So it's important to understand that some of these diseases may push the limitations of our new technology. And then understand moving forward, we need to think about resistance management, mainly not in the organic side of things, but on the conventional side, we really are dealing with 3 modes of action. And we already lost resistance in the frog eye system to the QOIs. So we need to think about how we're rotating our products and making sure we're protecting them for the long term. And so then here's Crop Protection Network, and a good resource of all sorts of tools available. And then here's my contact information if you have any other questions. So I have time for maybe some questions. Yep.
I was wondering if white mold is transferable in soybean seed.
It's not really getting in the seed, but if you harvest it and you have the little sclerotia there and you're not cleaning your seed, that's where I think it would be moving. You know what I mean? So making sure you're cleaning your seed so you don't have those, but that's where it's going to be collected in our systems. Just like tar spot doesn't go to the seed either, as far as we know, but if we don't clean our corn and there's still debris in there, we may be moving it that way as well. Yep. Okay.
So is it working? Yeah. So I have 2 questions and a suggestion for the corn growers here. So my first question is, I know you said you are not considering nutrition, but wouldn't it be an important thing to consider, since genetics also depend on nutrition, and the gentleman talked about soil health. But as an example or experience that we had at the farm, we have plants with some problems, and we grow several plants, but only few. And I remember I read before about the, I don't know if in English you say trophobiosis, the Francis Chabousseau, and also Ana Primaves in Brazil, they have talked about that a lot, that the importance of nutrition and soil health in order to prevent, because diseases are there, but it's how you manage the disease as humans. We have a room, some people get cold, others they don't. So the virus is out there. So the second question quickly, I know we have to go, is going back to the same case, and have you studied on your fields when you go out why some of the plants, they don't get sick, not all of them? And that would be also a good answer to get from those studies.
Right.
And lastly, about corn smut.
Corn smut?
Yeah, so.
We don't worry about it here.
Good, yeah, exactly, because it's delicious, eat it, it's a good profit, so economics.
So corn smut is one we got to injure to get that if we want it. So on nutrition, I would say most of our work we're trying to make sure we have healthy growing plants. But depending on the disease, that may, if we have a plant that's not as healthy growing, it may influence how that gets infected. I would say, for example, tar spot though, it's going after our best fed, best greenest corn. If we have droughty corn and it's not going well, we're less worried about that disease. It's really going after our healthy crop. So it's going to, again, depend on the disease. Some diseases may be influenced by reduced sulfur or other levels that the plant is missing in its normal growth. So again, most of our work is just looking under, we're making everything the same other than trying to push the disease pressure. So we want the crops to grow healthy. And then plant to plant, so whether we look at plants, so again, it's all going to depend where those spores are landing and how those spores are being dispersed. So if we think about white mold is where those spores landed, gained access, had an ability to get in. So it's not every plant, it's every, it may be the whole, every 10, there may be a section of 10 plants that are dead. I think it's where the pathogen's getting access. Some of our soil-borne pathogens, if they're not in contact with that seed and that right environment, then we may not have disease in this plant A and plant B may not have disease because the pathogen wasn't there to infect. So again, it's the host and the pathogen need to come in contact somehow first before the disease comes in control. Tar spot, generally once it's going, it's on every plant in my cornfields because we got spores that are just all over. So again, it's going to depend on each system how many plants we'll see. White mold is more, most of our soil-borne diseases, you have pockets, right, depending where the inoculum is in that field. So you know areas of the field where that's a bad white mold pocket, or that's where the SDS pocket is, because that's where those soil-borne pathogens are more readily available. And then we have the diseased plants, and we shed that inoculum back in that same area. It doesn't move as readily as compared to some of our foliar pathogens that are moving with their spores. There was a roundabout of not really answering any of that directly. But it's, again, each pathogen is going to have a different set of requirements that may make it worse or may make it better. And I know you can't just pick one. I understand that. So we're trying to look at one at a time. But I've been in fields where I have northern corn leaf blight, gray leaf spot, tar spot, and southern corn rust all on one leaf. Like, they can all happen at the same time. So then you have to think about, what is the battle I want to fight? What is the disease that's causing me the greatest concern? Maybe you're not worried about diseases. Maybe it's the other pests, it's weed management. You know, that's the target of, you know, what you need to think about for each field. Maybe one more question? Yep.
There's a theory out there that plants that are already sick are more susceptible to insect damage or disease, especially, or what's your thought?
And I think it's going to come back to what you're targeting. Because I think there's some that want, you know, a sick may open the door for other things to get in because we've already weakened the plant host response to whatever it's keeping it out. Whereas then there are others that maybe it's just, it wants the healthy plant. So it's going to depend on what the target is. Maybe insects see a dead and dying plant and they smell it and they want to go, easy access to a food source. So each one, and unfortunately there's not a good answer on that. So it all depends on what target we are. You know, if we have poor growing conditions, a wet field, we're more likely to have some of these seedling diseases and issues with water molds because we've got the environment that's now pushing the plants weaker and we've pushed the environment for that pathogen.